So, looks like we're populated. Hi, everybody. Good morning, good afternoon, good evening, wherever you are. Welcome to the 7th week in this fall iteration of the Long-Term Animal Research Seminar Series. This week, we hear from Dr. Faye Thompson and Dr. Kirsty Macleod. And we'll hear from Faye first, and before I introduce her, I'll make just a couple of quick announcements. If you are participating via Zoom, then you should see a Q&A tab that might be on either the top or the bottom of your screen. If you open that tab, you'll be able to type any questions that you have, as well as see and upvote other people's questions. At the end of the talk, we're going to go through those questions, starting with the ones with the most votes. But if you have a clarifying question that you feel like needs to be addressed during the talk in order to understand something, you can type clarification in capital letters at the start of your question, and Faye and Kirsty have both said that they'll do their best to answer those questions in real time. And lastly, recordings of the talks will be available on YouTube shortly after they conclude. So if you need to leave early or know others who are unable to attend live or just want to watch these a few more times, you can see both of these as many times as you'd like after they're complete. So we now have gotten to 12 o'clock. Welcome, everybody. I will introduce Faye first. So Faye Thompson is a postdoctoral research fellow at the University of Exeter. Broadly, she uses cooperative animal societies from insects to mammals as a testing ground for general evolutionary models of conflict resolution and cooperation. Her work integrates formal theory with long-term data and experiments, which allows her to investigate conflicts of interest within and between social groups and how these conflicts are resolved, thereby enabling this cooperation at higher levels of social organization. Her PhD and postdoc have focused on conflict within and between groups of cooperative banded mongooses, which is what we'll be hearing about today, I think, but she's also recently established a new termite study system that she's using to investigate the evolution of conflict, tolerance, and cooperation between distinct groups. So I'm really pleased that Faye is able to join us today, and I will turn things over to her now.
Just share my screen. Great, so thanks very much, Matthew, for that introduction, and thanks for the invitation to be part of this fantastic seminar series. So hi everyone, today I'm going to be talking about some of my work on the evolution of intergroup conflict in cooperative banded mongooses. So we're all familiar with cooperative species in which some individuals behave altruistically towards members of their group. And these species present an evolutionary puzzle because it's difficult to explain how cooperation and altruism can evolve in the face of selection for selfishness and self-interest. And classic research to answer this question has shown that factors that operate within the group, such as kinship and policing, can select for altruistic behavior. But favoritism towards fellow group members usually goes hand in hand with hostility towards members of other groups. And there's growing evidence that intergroup conflict can exert a powerful influence on within-group cooperative behavior. So in humans, theory suggests that warfare among ancestral bands has promoted the evolution of intense within-group cooperation, even among non-relatives. And in these models, intergroup conflict can, over many generations, select for genetic traits that amplify both within-group cooperation and between-group aggression. So investigating the relationship between intergroup conflict and cooperation requires a system that exhibits both behaviours, and banded mongooses provide just that. So the Banded Mongoose Research Project was started in 1995, and we've been continuously studying a population of banded mongooses in Queen Elizabeth National Park in southwest Uganda for the past 25 years. So we have a wealth of life history, behavioural and genetic data on over 3,000 individuals. So this provides a really powerful data set that we can use to test theories of social evolution. Banded mongooses live in mixed sex groups of around 20 adults plus their offspring. And in this species, there's low levels of reproductive skew among females. So once females reach around 1 year old, they start to breed. In males, there's an age-based cue for dominance with usually the oldest 3 males in each group monopolising access to females. And reproduction in each group is highly synchronized. So females in a group come into estrus within a few days of one another and give birth, usually on exactly the same day, to a communal litter that's raised by other group members. So there's really high levels of cooperation. Specifically, members of both sexes, typically not the parents, will babysit pups in the den in the weeks after birth. Females also seemingly indiscriminately suckle offspring. And once pups emerge from the den, they form an exclusive one-to-one relationship with an adult, again, typically not their genetic parent, and this adult escort teaches them how to forage and cares for them. So as well as really conspicuous levels of cooperation, banded mongooses also engage in intense fighting with neighbouring groups. And we know that these intergroup fights are highly aggressive and occur very frequently, and they're driven by competition for food, territory, and mates. And there's high survival costs for both pups and adults. So why has intergroup conflict in banded mongooses evolved to be so intense? Well, in humans, warfare has been rife throughout our evolutionary history. Collective conflicts are widespread and often highly violent and destructive. And a key reason for this might be that war is usually waged by leaders who can impose the cost of fighting on group members without having to experience any of the risks themselves. And these leaders can also gain a larger share of the benefits of fighting. So we wanted to understand whether this warmongering could also be a feature of animal contests too. So we developed a model that explores the more sinister possibility that individuals that initiate conflict can actually harm their fellow group members by exposing them to greater risks whilst not contributing to fighting themselves. We're terming this an exploitative model of leadership. And in our model, we extend the classic two-player hawk-dove game to pairwise contests of groups of size n. So a quick recap of the hawk-dove game. This is where contests between two players over a contested resource. Each player can either choose to play aggressive hawk or peaceful dove. Where a hawk meets a dove, it will always win the contest, so claims the entire share of the resource fee. Where two doves meet, they'll peacefully share the resource, so each gets a payoff of V over 2. And where two hawks meet, they engage in an escalated contest that each are equally likely to win. The winner claims the whole resource V, and the loser incurs a cost C, so the mean payoff for each player is V minus C over 2. So in our model, resources are contested by two groups of size n, and one individual in each group is assigned as the leader, and they choose whether their whole group will collectively play hawk or dove. And the payoffs to groups in our model are identical to the payoffs to individuals in the classic model, except that we allow for two forms of share of inequality. Firstly, in relation to the value of the resource, instead of the resource being shared equally amongst group members, such that leaders, here denoted by L, claim an equal share of the resource to each of their followers, denoted F, we allow for leaders to get a higher share of the winnings at the expense of each of their followers. And similarly, in terms of the costs incurred, instead of the costs being shared equally amongst group members, we allow for leaders to pay a lower share of the costs in relation to their followers. So these are results of our model for the situation where leaders claim an increased share of resources. The x-axis shows the cost of losing a fight relative to the value of the contested resource. So the section on the left is where the value of the resource is greater than the cost of losing, and on the right where the value of the resource is smaller than the cost of losing. The top panel shows the probability of playing hawk, and in the classic hawk-dove game shown in blue, where the cost of losing the fight is smaller than the benefit of winning, individuals will always play hawk. And as the cost of losing starts to exceed the value of the resource, then the probability of playing hawk decreases. And we see a similar result with our 2-group game. Here, the lines in red show leaders claiming an increasingly larger and larger share of the resource at the expense of their followers. And leaders continue to play hawk even when the cost of losing exceeds the benefit of winning. The bottom panel shows the expected mean payoff of the strategy. Again, the classic hawk-dove model in blue shows that the mean payoff never falls below 0. But our model shows that where leaders continue to play hawk, even when the costs of losing exceed the value of the resource, then the expected mean payoff for each of their followers can fall below 0. And this negative payoff can become increasingly large as leaders claim a larger and larger share of the resource, such that groups would be better off if the resource never existed in the first place. And we also see a similar result when we consider where leaders pay a decreased share of the costs. So again, leaders, as they incur a smaller and smaller share of the cost in relation to their followers, will continue to play hawk. And this will result in negative mean payoffs for each of their followers. So our model predicts that extreme levels of intergroup aggression will evolve when a subset of individuals can initiate conflicts that involve the whole group, and when initiators gain a disproportionate benefit or suffer lower costs than others. So we wanted to test the predictions of our model by using our data on banded mongooses and asking 3 questions. Firstly, do males and females experience different costs and benefits from intergroup fighting? Does the sex that suffers the lowest cost and the highest benefit initiate the conflict? And does this result in unusually severe levels of intergroup conflict? So to answer our first question, we looked at rates of intergroup mortality amongst adults in our population and compared mortality rates for males and females. And we found that males bear almost all of the mortality costs of fighting. So females are almost never killed during or as a result of intergroup conflict. So females seem to experience lower costs than males as a result of conflict, but do they experience greater benefits? So we know from previous work that intergroup interactions occur most frequently when females are in estrus. And we've observed estrus females lead their mate guarding males and the rest of their group into neighbouring territory and actually sneak off with rival males to mate during the chaos of fighting. And we know from previous work that pups that are born as a result of these extra-group matings, so pups are more likely to be born to extra-group fathers following intergroup interactions. And this is more likely to happen as groups become older and males and females within a group become more and more related to one another. So there's evidence to suggest that extra-group matings occur during intergroup interactions to avoid inbreeding. And we also know from previous work that these pups that are born to extra-group fathers are heavier at emergence than pups born to fathers within the group, and they also have higher survival to independence. So pups with extra-group paternity have higher fitness. So in order to test explicitly whether females gain larger benefits from intergroup conflict than males, we looked at whether the number of intergroup interactions experienced over an individual's lifetime predicted the number of extra-group pups that they have. And we found that for females here in orange, as they engage in more intergroup interactions over their lifetime, they have more extra-group pups, but this relationship doesn't hold for males. So we can see that females can enjoy fitness benefits from intergroup fighting via extra-group paternity that males do not. So in answer to our second question, do females, as the sex that suffer the lowest cost and obtain the highest benefit, initiate conflict? So to look at this, we examined whether the estrous state of focal and rival groups in an interaction, whether they were in estrus or not, predicted the number of intergroup interactions that they engaged in. So if females are the sex to initiate conflict, then we might expect a pattern like this, where females, where we observe the highest number of intergroup interactions when focal females are in estrus, irrespective of the rival state, the estrus state of rival groups. However, if males are the sex to initiate conflict, then we'd expect that we'd observe the most intergroup interactions when their own focal females were not in estrus, but rival females were. And we found that our data most strongly supported the prediction for females being the sex that initiated conflict. So intergroup interactions occur when females are in estrus, irrespective of the estrus states of the rival group. So does this result in unusually severe levels of intergroup conflict? So previous work by Richard Wrangham has compared mortality rates for populations of chimpanzees with that of subsistence farmers and hunter-gatherer human societies. And they found that chimps and humans have similar levels of mortality from intergroup conflict. So we wanted to compare our mortality data for banded mongooses to see whether banded mongooses have similar rates of mortality. And we found that intergroup conflict in banded mongooses is as intense as chimpanzees and some human societies. And this doesn't appear to be a trait of social mongooses. So we looked at comparable data from the Kalahari Meerkat project and found that meerkats have relatively low levels of mortality from intergroup fighting. So to sum up, banded mongooses experience high levels of mortality from intergroup conflict, and females are the ones to initiate this conflict for genetic benefits, but males are the ones that bear the cost. So this fits our exploitative model of leadership. And our theoretical model and our data suggests, therefore, that destructive warfare can evolve when there is a decoupling of leaders from the costs that they incite. So thanks very much to everyone that's been involved in this research and the project for the last 25 years, particularly our amazing Ugandan field team, without whom none of this would have been possible. And thanks very much to you for listening.
Oh, that was terrific. Thank you very much. I have a lot of questions. I'll start with one that I'm guessing others are also thinking, which is, do males have a way of preventing females from initiating conflict? Are there any counter strategies to this, since it seems to be somewhat costly for males?
Yeah, not really. When females are in estrus, the males that are mate guarding them follow them nose to tail all day long. So really, where those estrus females choose to go, their males will follow them, and the rest of the group. Mongoose groups don't split up and forage separately. They're very cohesive. They stay as one group. So really, if females decide to move into neighbouring territory, then really the whole of the group can follow them. And it seems like there's very little that males can do to avoid that, because they're so driven to mate guard their females, and they aggressively defend their females from other males within their group, and also from other males from rival groups trying to mate with them.
Yeah, so that's interesting. So I'm thinking about costs and benefits, and in that figure that you show with the expected net cost to the followers, I'm thinking about how that can evolve. So I'm wondering if you've thought about how to kind of, it seems like the cost of abandoning a mate guarding session must be greater to males than the risk of death. Have you quantified that in any way to try to compare those?
No, but that would be really interesting to do. So yeah, to look at the opportunity cost, I guess, of giving up a mate giving up your female in order to avoid the mortality costs of fighting. No, we definitely got the data to do that, so that'll be really interesting to look at.
We also have a question here from David Fisher, who asks, do females lose inclusive fitness by causing costs to related males when they go off to war, essentially?
So I don't know is the answer to that. But I guess we could look at that as well, because we've got the genetic pedigree for our population. So yeah, we can look at kind of the cost that females, sort of the indirect cost that females lose from their males being killed during fighting.
Well, let's leave it there for now. I thought that was really interesting. Terrific to see such exciting new theory applied to such a rich data set. So thanks very much.
Thank you.
Our next speaker today is Kirsty Macleod. She is a Marie Curie Actions Research Fellow at the University of Lund. Kirsty asks a range of really interesting questions about the role of stress in animals' lives, where it comes from, how it affects them, how they respond to that stress, and how sociality and relationships can either add to or reduce it. She's investigated ecologically relevant stress, especially predation stress, in a range of species from meerkats and snowshoe hares to fence lizards and tree swallows. This range of study systems has allowed her to be able to ask similar questions using different methods and at different levels. And the result so far is a really nice body of work that gives us new insights into the proximate and ultimate implications of stress, maternal effects, and sociality. So with that, I'm happy to welcome Kirsty, and I'll turn things over to her now.
Thanks. All right, I'll get this set up. Okay. Hi, thank you very much to Matthew for that very kind introduction, and to Matthew and Liz for the invitation. So as Matthew said, I'm going to talk today about stress and survival in vertebrates. And I'm especially, oh, hold on. Just need to, there we go. Yeah, I'm really thrilled to be a part of a seminar series focusing on long-term animal research, because this is where I started. So this is very much where my heart lies. My PhD investigated the cross-generational social interactions, specifically cooperative nursing behavior and their long-term implications for individuals in Kalahari meerkats, which Faye mentioned. So I've continued to be primarily interested in social interactions within the family and their long-term impacts for individuals and groups. But my work now focuses on the effects that mothers have on their offspring, and particularly the ecological and evolutionary consequences of these effects. So we know that mothers pass on information to their offspring through DNA, but the environment that the mother is in and her own phenotype can also affect offspring through non-genetic means. And these are called maternal effects and are increasingly thought to be important in ecology and evolution. So for example, effects of the mother's condition or diet or age. So my work particularly seeks to answer two main questions. First, do maternal effects like this affect fitness in natural systems? And how do maternal effects vary across contexts? I think I'm really driven to answer these questions, as I think that understanding maternal effects and their importance in natural systems will help us to better understand our own reproductive health, the process of evolution, and potential downstream effects of the major environmental changes that we're currently experiencing. And today I'll also argue that a better understanding of the importance of maternal effects could be important for our interpretation of patterns in long-term data. So I primarily investigate these questions through the lens of maternal stress as a particular maternal effect of interest. And stress provides a useful framework for testing the importance of maternal effects. Firstly, because stress is experienced across taxa in broadly comparable ways in terms of types of stressors that animals experience and the physiological effects that those stressors have. And these physiological effects are relatively well studied also across taxa. And lastly, as I alluded to before, we live in a rapidly changing world, and increasingly impose and create novel stressors for wildlife. So studying different ways that stress can influence individuals and populations is therefore also increasingly important and relevant. So what do I mean when I use the term stress? This obviously has anthropogenic connotations, and many of us may have different ideas about what that means. So I'll just lay out how I'm going to use that term. So organisms, including humans and most vertebrates, work to exist in a state called homeostasis. And this is just a state in which physiological parameters such as blood pressure, blood glucose, etc., are functioning within narrow life-sustaining ranges. So stimuli that disrupt homeostasis are what I'll refer to as stressors. And this period of physiological imbalance that they induce, I'll refer to as being under stress or stress. So stressors come in a variety of forms, such as predation risk, exposure to pollutants, the sort of conspecific aggression that Faye was talking about, food restriction, and of course also what the majority of people listening may be more familiar with, giving a talk, although it's slightly different when I'm saying here rather than standing up in front of you all. So this variety of stimuli, despite the diversity, have common physiological outcomes. So initially, the stressor is perceived by the brain, and then this triggers an endocrinological cascade, which I won't go into in great detail, except to say that one of the results is the production of glucocorticoid hormones. And these are simply metabolic hormones. They're a part of everyday functions across vertebrates. So they're part of a daily cycle that we all also experience. So they're often referred to as stress hormones, but they actually have a number of roles in the body which are far more benign. But in the context of the physiological stress response, these function to promote processes that return the body to homeostasis. So with this in mind, today I'm going to explore two main themes related to stress. First, how does stress affect the survival of adults and their offspring? And what are the consequences potentially for populations? And also some possible mechanisms for these effects. So I'll be describing two different studies, one looking at snowshoe hares and then one on eastern fence lizards. And secondly, how do the transgenerational effects of stress vary based on the environments of both the mother and the offspring? And I should have enough time for also a sneaky part 3, which is going to be more of a sort of trailer for some current work that I'm doing, which follows on from these two themes. So first of all, many of you will be familiar with the snowshoe hare from the very famous snowshoe hare lynx population cycle, which you can see a version of at the bottom of the screen there. So this has been used for many decades as a classic example of predator-prey dynamics. So just a very brief description, although I'm sure you're all, you've heard of this before. The hare is a key prey item for lynx in this system. So as numbers of hare increase, this increases food availability for lynx, and their numbers then also increase. But this increased predation pressure then causes a crash in the number of hares, so that their numbers go down, and the lynx numbers, as you can see, tend to follow very closely. And this has been studied for many, many decades, and these are very consistent patterns. So it's of course intuitive that predation of the hares by the lynx is a main driver of these patterns. But for every encounter between a lynx and a hare that results in the hare's death and consumption, there are numerous encounters where the hare either manages to perceive cues from the predator and evade detection by hiding or changing their behavior in other ways, or where there's a chase and the lynx is not successful in catching the hare. So both of these encounters are obviously likely to cause stress for the hare and the triggering of that physiological and endocrinological cascade that I described. So we wanted to know, could this also potentially contribute to this cycle that we see in the long-term data? So to test whether the stress of predator encounters affects survival, we conducted a predator exposure experiment using wild pregnant female hares in the Yukon in Northwest Canada. And this is work that I did with Michael Sheriff at Penn State. And this was using data that he collected several years previously. So how this experiment worked was that, as I said, these wild hares were trapped in the Yukon. They were captured at the stage of mid-gestation, and then they were assigned to either a predator exposure treatment or a control treatment, and they each had individual pens. So individuals in the predator exposure treatment were exposed daily to a simulated predator, which in this case was a live trained dog. So the dog was walked through the pen each day, and it wasn't allowed to come into contact with the hare, but the hare was obviously able to get any auditory, physical, chemical cues from the predator. So it was aware of the predator, but it couldn't actually be killed by the predator. And in the control treatment, a handler was walked through the enclosures. So that was just to account for any potential effects of exposure to a human. And this continued from capture, so again, that was mid-gestation, until the females gave birth. And then the treatment was stopped. So any effects on offspring were solely through effects that had been on the mother, not on the offspring. So maternal and offspring survival were monitored to weaning. So I'll first show you the results that my analysis found for adult female survival in response to the predator exposure treatment. So on the y-axis is proportion of adult female survival, and you'll see data from 2 years. It was replicated over 2 years, and the predator exposed treatment is in the pale bars. So as you can see, although the predator was not allowed to come into physical contact with the hare, it nevertheless had strong effects on their survival. So there was actually 100% survival in the control group over both years, but predator exposure without actual predation reduced survival to about between 60 and 70% in both years. But what was really interesting was that we found some really interesting results looking at how predator exposure of the mother affected survival of the offspring. So remember that offspring were not themselves ever exposed to the predator. So here we have the number of live offspring that were produced between the control and predator exposed treatment. So first, there were no differences in the number of offspring that were born to mothers in either treatment. And that's not surprising because treatment started in the middle of gestation, so probably too late for any resorption of fetuses or anything like that. But between birth and weaning, the survival of offspring in the predator exposed group was massively reduced, such that females that had been predator exposed produced an average of 0.1 offspring per surviving female, which is obviously not very many offspring in this group. So why are these effects potentially important? So just to illustrate this, if we look at the different group sizes in the treatment, initially the predator exposed group was actually quite a bit bigger. But then when you account for differences in the number of surviving females, and particularly in the number of offspring that were weaned, so when I say weaned, that's when they are independent of their mother and no longer eating solely, getting nutrition solely from their mother. The final group size was really different. It was completely reversed. So the predator exposed group decreased by about 1/3, while the control group increased by over 100%, which is obviously what you'd expect during a breeding cycle. So if we return to the hare lynx population cycle, it does indeed seem possible that the stress alone of predator exposure could also be contributing to hare declines in addition to actual predation events. So in other words, by focusing on this relatively short-term cross-generational interaction, we can fill in some detail regarding patterns that have been evident in the long-term data for a long time. And this study illustrates that there is potential for population level effects of transgenerational stress at this level. But what we don't get from this study is an indication of what the mechanism is for these dramatic effects. And that's where I turned next. So we know from previous study that there is a group of hormones that are increased in response to stress in basically every species. So as I mentioned before, that's the glucocorticoid hormones that are produced as a result of this endocrinological cascade in response to stressors. So just a few examples. Social stress in meerkat groups leads to substantial elevations of glucocorticoid hormones in the blood. Food restriction in rhinoceros auklet chicks leads to high levels of glucocorticoid deposition in the feathers. And in the Fijian ground frog, the sight alone of a cane toad predator induces increases in glucocorticoids that last for hours after the encounter. So I was interested to know if it's potentially glucocorticoid elevations that reduce adult survival and reproductive output in response to stressors, such that we saw in the hares. But to answer this question, I turned to a different study system, the eastern fence lizard, Scalopus undulatus, which is a really great system in which to answer this question. And again, this work was done with Michael and with Tracy Lankilder, both at Penn State, or they were both at Penn State at that time. So the fence lizard is a really great system for these questions. Firstly, because it's possible to non-invasively manipulate glucocorticoid hormone levels by applying it directly to the back of the lizard. It's a lipophilic hormone, so it absorbs easily through the skin and scales, which means that we don't have to use an implant or an injection, which itself can cause stress. And it's just so much easier. It makes it much easier to do on a kind of bigger scale. So the holding of the lizard is actually just purely for photographic purposes in this case. You don't need to do that at all. You can just pipette it onto the top of the lizard, so it's very low disturbance. And secondly, the effects of environmental stressors and how they impact glucocorticoid levels naturally have been really well studied in this species, thanks to Tracy's really impressive body of work, particularly looking at interactions between fence lizards and invasive fire ants in the southeast of the US. So we're able, thanks to this work, we're able to recreate the physiological effects of a stressor that they actually encounter in the wild. So many of you may be familiar with the invasive fire ant. This is an ant that was unintentionally brought into the southeast of the US, I think via fruit cargo, and it's very rapidly established a stronghold and has spread up, I'm sure, through North Carolina. They are horrible little critters, and if you've ever been bitten or stung by one, it becomes easier to understand how such a tiny predator can very quickly overwhelm and kill a lizard. It really doesn't take many of them to do that. But obviously, the lizards have evolved behavioral mechanisms to avoid being killed, but that means that they are subject to regular non-lethal attacks. And those attacks result in an increase in circulating glucocorticoids, which this topical glucocorticoid application protocol can easily mimic. So colleagues and I conducted an experiment to test whether elevation of glucocorticoids at this ecologically relevant level affects survival and reproduction. So to do that, we caught pregnant females in the wild at 3 sites in South Alabama, where fire ants have established a very strong stronghold. So this is somewhere where the lizards are constantly interacting with fire ants, and so having a stressor at that level daily is a very realistic encounter and level of stress. So we caught pregnant females in Alabama, and then we brought them into the lab where they were split into control and treatment groups. So the treatment group received a daily dose of glucocorticoid hormone, corticosterone, which I'll refer to as CORT. This is the primary glucocorticoid in reptiles as well as birds and lots of other groups. So this was dissolved in oil, and that was used as a vehicle to be pipetted onto the lizard, and the control group just received the oil on its own. And females received these treatments from capture, which again was about midway through the gestation period until they laid. So this is just, again, just to show that this dose protocol, it just results in a short spike of glucocorticoid elevation. So it's not leading to a sustained chronic level of glucocorticoid elevation, which can easily have pathological effects. So that's just to emphasize that this is just a very small spike. So we really wanted to try and recreate stress at a very ecologically relevant level. So we incubated the eggs, we monitored adult survival, and then we also monitored how many eggs hatched successfully of those that were laid by the females. So did we see similar results using our direct glucocorticoid manipulation as we did when we exposed hares to natural predators? And again, this is showing proportion survival, and again, this was replicated over 2 years. And you'll see that, yes, the pattern was very similar when we directly manipulated glucocorticoids. So over both years, the glucocorticoid treatment during gestation significantly reduced adult survival. And this effect was particularly strong in 2016. And I'll come back to that. So moving on to egg hatching success, again, we saw similar reductions in offspring in clutches from cord-treated mothers, again, over both years. And again, the effects were particularly strong in 2016. So returning to our initial set of questions, do maternal effects have the potential to affect fitness in natural systems? Well, in both of these studies, we showed that maternal stress effects, either through exposure to a natural stressor in the hares or a direct manipulation of the stress response in the fence lizard, did influence adult survival and reproductive output in remarkably consistent ways. And this is obviously likely to contribute to overall fitness. But to better understand the generality and importance of these patterns, we need to understand how consistent they are across natural contexts. So what do I mean when I say that? So as an example, the same amount or level of stress experienced by an animal may cause different degrees of physiological response depending on the environment in which the stressor is encountered. So as I flagged earlier, and as I'm sure you would have noticed anyway, the effects of our experiment were really much stronger in 2016 compared to 2015. So I'm just showing the data for adult survival here, but it was really the same in terms of hatching success. There was a significant drop in both in 2015, but in 2016, this was really, really striking. So this was despite the fact that the protocol was identical and the females, although not the same individuals, were drawn from the same population. So we were really scratching our heads about what the difference could be between these two breeding seasons. And then we thought a bit more literally about what's actually between these two breeding seasons, which of course is winter. So we hypothesized that perhaps there may have been some difference in the overwinter conditions that resulted in females then experiencing the same level of stress, but having a vastly different physiological response. So that would be sort of the equivalent of having to undergo some physical activity after a good night's sleep versus a very poor night's sleep, and then that resulting in different outcomes. So winter is important for reptiles because they undergo, even in the relatively warm deep south of Alabama, they undergo this brumation period, which is sort of like going into energy saver mode. It's a time where there's relatively little food availability, and also it might be warm enough, but it's not necessarily warm enough in order to metabolize the food if they are able to find any. So we were interested in looking at temperature differences between these two winters to see if perhaps a warmer winter meant that they were under greater metabolic strain because they were coming in and out of torpor. So this is a time series graph here just showing the maximum and minimum daily temperatures across to the end of 2014 to the middle of 2016. So these are the breeding season periods in which we conducted this experiment. spring and summer of 2015, and then the same in 2016. So we were interested in comparing these two overwinter periods. And as we expected, we actually did find that the winter prior to the season, where they really seemed to be hit very hard by the stress treatment, this was significantly warmer. So this is anecdotal, but I think, well, it's got me really interested in the potential for overwinter warming to really be an aspect of climate change that's particularly detrimental to reptiles, especially. And I think it's a really neat area of potential future research, and I think it will be increasingly important in understanding how animals respond to stress. So in addition to variation in stress effects based on the maternal environment, the fitness outcomes for offspring may also depend on the environment that they experience during their early life. So, for example, if we imagine that a mother under stress produces offspring of a certain phenotype, and commonly that's thought as a generalization to be smaller, more timid offspring, in a benign environment, this will probably lead to reduced survival. For example, if they're outcompeted by larger conspecifics, if they are less exploratory and as a result forage less or have lower foraging success. But in a high-risk environment where all individuals probably have to forage less, a smaller offspring may fare better due to reduced energy requirements, and its timidity, as an example, may mean it's less likely to be predated in a high predation environment, or other aspects of its behavior could result in increased survival in high-risk environments. So this is a sort of generalization of the environmental matching hypothesis, which suggests that mothers might adaptively program their offspring to fare better in an environment that matches what they have themselves experienced during gestation. So this does assume that the maternal environment during gestation has some degree of correlation with the environment that offspring experience. So returning to the fence lizards, we do know that maternal stress during gestation has effects on offspring phenotype. This is work that was done by a colleague, David Ensminger. So he found that the same protocol that I described resulted in offspring that were smaller, more risk averse, and had lower baseline glucocorticoid levels. So we wanted to know, do these changes in offspring phenotype actually result in different fitness outcomes? So do they change survival of offspring? And we also wanted to know, do these fitness differences depend on the environment that they find themselves in early life? So we replicated the same maternal stress experiment, where we treated the mothers with glucocorticoids during gestation and then followed their survival and their hatching success. But this time after birth, we relocated offspring to outdoor naturalistic enclosures in Alabama, which mimicked either a benign or a stressful environment. So in this context, this meant we set up low predation versus high predation environments. So we did that by either removing fire ants from these large outdoor enclosures or allowing them to persist at naturally high densities, which in Alabama, which is actually where the fire ant came into the US through the port of Mobile, so density is very high there. That we left fire ants in the enclosures basically to create these kind of lower high predation environments. So I just have a short video here that'll just show you what this looked like. So yeah, we had these large 20 by 20 enclosures. They all had equal amounts of shelter and so on. And we essentially did a census every day. So we walked through, there's a tiny marked lizard there. And we monitored survival, detectability, other aspects of behavior, substrate use, and so on. There's a spider. Sorry for my very shaky camera work, but that's just to give you an idea of the census and the enclosure types that we had. So I'm just going to talk about the survival stuff today, but there were some quite cool differences in other anti-predator behaviors that I could perhaps talk about as well later. There's another little marked baby. Okay, so yeah, we were essentially investigating two competing hypotheses with this data. So first, if maternal stress effects are always maladaptive and fitness effects crucially do not vary depending on the environment that the offspring find themselves in, then we expected to see reduced survival regardless of environment type. So offspring from court-treated mothers should have lower survival in both benign and stressful environments. Alternatively, if the fitness outcomes of maternal stress depend on the offspring environment and whether it matches that of the mother, then we would expect that offspring from stressed mothers should do better in high risk, or in this case, high predation environments. So if the smaller size and greater risk aversion perhaps better suited them to high fire ant predation, then we would expect to see that as a survival outcome here. So we monitored survivorship over 10 days, which has previously been shown to be a particularly vulnerable period for fence lizard offspring, particularly in the context of fire ant predation. So what you'll see here is a kind of traditional survivorship curve. Obviously on day 1 post-hatching, that's the day of birth, then you have 100% survival. And then they went into enclosures at that point, and then you'll see it drop off after that. So low predation environment is in black and high predation environment is in red. So I'll first show you the results for offspring from control mothers, which are in solid lines. So there was a pattern for reduced survival in high density fire ant enclosures, which is what we expected. So there was initially a sharp drop off in survival, and then it flattened out. But it flattened out at about 80% survival, which was actually much higher than we expected. So this was not a significant difference. And then if we add in the offspring from court-treated mothers, there was no difference there either between environments. And overall, there were no differences according to any of these treatments. So essentially, we did not see effects of the prenatal stress treatment on survival or an interaction with the environment of the offspring. So again, returning to our initial questions, how do the outcomes of maternal effects vary across contexts? Well, in eastern fence lizards, there was some evidence that the environment in which mothers experience stress influences outcome. But we didn't find any support for the environmental matching hypothesis in offspring or any kind of context-dependent survival effects. And this, I think, suggests that we should just be cautious in how we interpret the long-term importance of phenotypic effects in offspring, especially for fitness relevant traits. So as I mentioned, we did find phenotypic differences in offspring, but when we actually then put them in a kind of a real world scenario, it didn't actually have any important effects on survival. So I think I have heaps of time. So for the last part of my presentation, I'm going to talk a bit about what I've been doing more recently as part of my current fellowship, which follows on really closely from this work and these questions. So I got my start in social species, and I'm really fascinated by the diversity of postnatal associations between mothers and offspring across species and across taxa. So in fence lizards, of course, mothers just lay their eggs and then they never see their young again. But in many species, mothers that care for their young or even are just near their young during early life have the potential to really be able to influence the outcome of parental effects. So my current and future work seeks to integrate maternal stress and the social environment and asks two questions in particular. First, do postnatal associations change the outcomes of prenatal stress effects? So, for example, if a mother that was stressed during gestation then provides a lot of care after birth, are they then able to buffer the effects of the prenatal stress on their offspring? Or alternatively, if a mother remains stressed after birth, and then provides really crappy care to the offspring, does that perhaps exacerbate the effects of the prenatal stress environment? And I'm also interested in how parental stress effects potentially contribute to social structure. So, for example, if prenatal stress alters the offspring's propensity to be affiliative or potentially to disperse. So I've been testing these questions in another wonderful lizard species, Lyophilus whitei, or the white skink. And this is a skink that's native to southeastern Australia. And again, it's a lovely system for testing these questions because we can manipulate glucocorticoids in much the same way as I've described in fence lizards already. But as you can already tell from this picture, they're a quirky species, less so in Australia where there's a lot of social reptiles, but relative to reptiles in the rest of the world, they're fairly unusual because they're facultatively social. So adults live together in long-term associations in burrow systems, and offspring they'll fare fine if they go off on their own immediately after birth, but very commonly they remain with their parents. So that provides a really nice system for the sort of work that I want to do, because it's possible to manipulate the postnatal environment in a way that's ecologically meaningful. So I can create kin groups, but also single-housed offspring immediately after birth, and then test the effects of the immediate postnatal environment. So I'm at the moment mostly trying to answer this question: does associating with the mother during early life alter the phenotypic effects of prenatal stress? And this is work that I'm doing with Jeff Weil at the University of Tasmania and Tobias Uller, who's at Lund. So I don't have any results yet to show you from this, but I'll just walk you through the experiment because I'm really excited about it. I think it's going to be really cool. So again, I replicated the experiment that I described in the fence lizards in Lyophilus. So manipulating glucocorticoids in females that were caught again during mid-gestation until they give birth. So a primary difference between Lyophilus and the fence lizards is that these are live bearing, which of course facilitates those kind of postnatal interactions between mothers and offspring. So they give birth to these completely ferocious babies. And I got morphological measurements from all these babies immediately at birth to look specifically at the effects of prenatal stress. And we're also collecting brain tissue samples, which ultimately we'll use to look at differences in the expression of genes related to stress phenotypes. But then the second part of this experiment was housing offspring either with their mother, as you can see in the top right there, or singly housing them in kind of little baby bachelor pads. And this treatment went on for 3 weeks, during which time I monitored their behavior and their growth with or without their mum. And again, got morphological measurements all along that period, and also more tissue samples, so we can look explicitly at the prenatal treatment and the interaction with the postnatal social environment. And this part is really cool. We then were able to put them again into these big outdoor enclosures at the University of Tasmania. And we were able then to look at how the interaction of these two treatments affects social behavior and dispersal and survival. So here's Jeff laboring away under the hot Tasmanian sun. If anybody ever says to you, let's do a lizard experiment with large outdoor enclosures, definitely run a mile unless you have a brilliant team of people, because they're such a pain to put up. But I think they enable us to answer some really cool questions. And this was done with Alex Buffet-Hall, who's a brilliant PhD student of Jeff's, and she put in many, many hours of collecting really, really fine scale data, looking at how they were moving around in these enclosures. So this is a GIF that she generated. So here you can see this. So this is the mother and her two offspring. And of course, this was all blind, so I don't know which one she was associated with after birth. But we were really interested in, we created these dispersal corridors, so only the offspring could get through these mesh pits. So this guy is obviously quite exploratory. He's headed straight over there, isn't spending much time with his mom. his or her mum. And this one is kind of testing the water, but then ultimately goes back and associates with the mother. So yeah, we have details on all the families, and yeah, none of this is analysed yet, but just to give you a flavour of why we're interested in this sort of thing. Alex has been able to create social networks, both for the families and for these kind of mini populations within each enclosure. And then we've also been able to create things like home ranges and degree of home range overlap between offspring and mum. And this is really cool in the context of Jeff's long-term data. So this is a map of Jeff's long-term field site in southeastern Tasmania. So he's been collecting data here throughout the year and throughout the breeding season for over 10 years. It's a really elegant system. He's shown that pair bonded males in black and females in white live together in long-term burrow and crevice systems. And also the offspring show considerable variation in their dispersal patterns. So the green dots are offspring that remained with their parents until at least the following year. And then red indicates the dispersers, and you can see there's lots of variation in who's going where. So we're really interested to see with our maternal stress experiment whether maternal effects may play a role in dispersal dynamics of populations like this. So Jeff and I are also collecting blood samples from adults in the wild to determine whether natural variation in stress response profiles contributes to patterns arising in the long-term data. So we can sample females at these sites in the wild throughout the breeding season, and then next year we can go back and see where their offspring are. So I think that's super cool. I'm very excited about that. So I hope I've managed to persuade you that there's lots of scope for maternal effects and stress effects in particular to contribute to large-scale population processes and life history dynamics. So for example, through effects on survival and reproduction in the hares and in the fence lizards, and that glucocorticoids are likely to play a role in these. And yeah, watch this space for potential impacts of the postnatal social structure on these dynamics as well. So obviously this was done with a huge number of people and collaborators. So the hare and fence lizard work was all with Michael and Tracy at Penn State. And the hare stuff obviously is part of the long-term system there with Charlie Krebs and Rudy Boonstra. And the Lyophilus skink work is all with Tobias and Jeff as part of my Marie Curie fellowship. And huge field teams helped out with all of those, particularly the construction of outdoor enclosures, as I said, which it really takes a village to do that stuff. So I'm very grateful to all of these people. So I will leave it at this slide if I can. Do you want me to stop screen sharing?
It's up to you. Either way, if you want to be able to refer back, you can keep sharing.
Okay, sure.
I thought it was really terrific. I have a couple of questions while we wait for other people to send theirs in. Focusing on the hare lynx dynamics. You know, if you didn't know that lynx actually killed hares, right? And all the information you had was the studies that you've done. I don't know, I might look at that and say that was adequate to explain the pattern that is shown in that figure. So I guess my question is, what proportion of this cyclical population dynamic do you think, if I'm going to ask you to speculate pretty wildly here, but what proportion of that dynamic do you think might be explained by the stress that?
Yeah, I think that's a great question, and the answer is of course that I don't know. I don't think it's common to find hares that have just dropped dead throughout the Yukon because of stress. I think it's probably more likely that what the results are in the wild is that it just leads to animals that are less able to cope with future stressors, and that I suppose it may depend slightly on the level of predation risk at the time, because obviously that fluctuates wildly as well. So when there's very high predation risk, it might be that animals that have experienced considerable stress in the past then become less able to cope with future encounters with predators. So I think that's a really good question, and I think it's very hard to parse out, basically. But yeah, I would say I'm not sure that there's many that are just dropping dead. I think it's probably more likely that the differences in reproductive success are contributing more.
And my next question is about that difference in reproductive success. Do you think that's about differences in maternal allocation or something that's happening after birth, or is it an inherited epigenetic difference? What do you think is causing that?
Yeah, that's also a great question. And I think it's more to do with postnatal allocation in terms of caring for their offspring. That would be my guess, but again, I think that would be a really cool thing to go and follow up on.
So I have a question about the fire ant lizard dynamics. How variable are the fire ant population dynamics such that like an offspring would be likely to find itself in a different fire ant stress environment than its mother?
They are, in this part of South Alabama, you really have to watch where you step because there's so many fire ants there. So I think in terms of the correlation between the maternal fire ant risk environment versus the offspring, I think it's likely to be quite similar. I think something that's really cool about this system, and we've just submitted a paper on this, and so hopefully this will be out soon and this may answer some of your questions, but I think what's cool about this system is that there are a whole range of different things that eat fence lizards. So they're subject to predation from birds and raccoons and snakes and ants. And the behaviors that we saw that differed, because offspring did actually differ when we looked at the behaviors. So they really use their environment very differently. So offspring from stressed mums tended to kind of stay low to the ground and be a bit more cryptic, which totally makes sense as an anti-predator strategy if you are avoiding a bird, for example, but not avoiding a fire ant. So there's this potential cross-selective pressure of avian and other predators. I think I've strayed quite far from your initial question, but I think basically the predation environment is probably quite correlated, but I think the really interesting thing there is that there's totally different selective pressures, and I think that could contribute to why we don't see any effects on survival, is that they may be responding to a historical predator rather than a novel predator.
Thank you. Very cool.
And we've had a couple questions come in here. One from Christine Dre, who asks,
Hi, Christine.
She asks, are you looking at any other physiological measures related to court? I think either in the completed work or in the ongoing enclosure work.
Yeah, I would really like to. So in the current stuff with the skinks, we have blood samples and brain samples, and yeah, obviously court is just one part of the stress response. We do have a bit of a tendency to focus on it in the stress literature because it's very stable, it's easy to study, it's easy to pick up in blood samples. But yeah, I'm really interested in other potential factors as well. And I'm hoping that using the tissue samples to get at the gene expression differences will also help to kind of round out that picture. So yes, hopefully with the current stuff, we'll be able to kind of broaden that out.
All right, and let's close out with one more question from Eli Strauss, who asks, do you think you would see the same survival result that you saw in the predator-exposed pregnant hares in non-pregnant hares?
That's really interesting as well. Yeah, that's a great question, because I think pregnancy puts obviously a lot of stress on individuals, and it means that yeah, their response to stress could be really different. So yeah, I don't know the answer to that, but I'm really interested in the idea of just how additive physiological stressors may interact, and I think pregnancy is a really neat one because it has quite often consistent effects on things like immune function and metabolism. So yeah, that would be really cool. Thanks, Eli.
Great. Well, I think let's leave it there. Thank you to both of you for two really thought-provoking and interesting talks. And thanks for giving them. Christine Dre, by the way, says, hi, Kirsty, great job. And Emily Levy says, amazing talks, both. Thank you. So thanks for both of you for joining us. And for everyone else out there, we'll see you in the same place, same time next week. Thanks very much.
Thanks for having us.